Hi, I'm Ivan. As an LSAMP research fellow mentored by Dr. Ho at the University of Texas at Tyler, I studied how environmental changes impact microbial fitness. Bacteria frequently encounter changing environments, such as within our gut microbiomes, where trillions of bacteria help to maintain homeostasis by breaking down different nutrients. Understanding how bacteria adapt to fluctuating environments can lead to advancements in evolutionary biology and public health. When bacteria encounter a changing environment, dioxic shifts allow them to switch between favorable carbon sources consumed for growth. This study investigated E. coli populations transferred to glucose minimal media after evolving in nutrient-rich Luria bertani, LB broth, for 900 days. We hypothesized that the evolved populations would exhibit slower growth in glucose minimal media due to dioxic shifts. To test this hypothesis, I completed a series of competition assays between evolved populations P203, P215, P220, and a wild-type E. coli population. Additionally, I competed a non-evolved ancestral population 0 versus the wild-type as our standard of control. Each population was revived and inoculated into both media and then incubated at specific time points. Serial dilutions, platings on TA agar, incubations, and colony counts were performed at each time point. Each assay was performed 3 times and data was analyzed using RStudio. Figure 5 shows growth curves in LB broth, where the x-axis represents time and the y-axis represents colony-forming units per milliliter. These graphs show that the evolved populations, which are purple, generally match the wild-type populations, which are pink. Figure 6 shows growth curves in glucose minimal media, where the x-axis represents time and the y-axis represents colony-forming units per milliliter. These graphs show that the evolved populations initially grew faster than the wild-type populations, but at the 6-hour time points, the wild-type population started growing faster than the evolved population, thus confirming our hypothesis of dioxic shifts in glucose minimal media. Figure 7 shows relative fitness values in both media. P215 had a slightly higher relative fitness in glucose minimal media compared to LB broth, but P203 and P220 had a lower relative fitness in glucose minimal media compared to LB broth. In summary, our findings highlight dioxic shifts and differential fitness outcomes of the evolved populations after transferring between environments. Future studies will focus on continuous exposure to glucose minimal media and genomic analysis to uncover specific adaptation processes. I would like to thank LSAMP for supporting this research.